everybody, my name is Mara Burrows. Today is April 5, 2020, and this is for my Bio 225 class, week 4, lab 6. Today we're doing the biochemical testing for microbial identification. We're gonna do the methyl red testing, the Voges, Proskauer, and catalase testing. I've already incubated my MRVP broth, one with E. coli and the other one with S. epidermis. As you can tell, the inoculated broth is cloudy. Same with the E. coli broth. I have my test tubes in hand. I have already dipped them in alcohol and they are pretty much dried off already. I've labeled 2 E. coli and 2 S. epi. I have my test tubes labeled. There is S. epi on this side and E. coli on the other. These are not the very best test tube holders, but it will do. Okay, so I'm gonna pour the E. coli into the test tubes half and half. Just shake that up. Half on this tube. And the other. That is there, and I'm also gonna pour the S. epi on the other two, half and half. I'm gonna actually move these test tubes out of the way. Okay. Next, what I'm gonna do is I'm gonna go ahead and take my methyl red and I am gonna do 7 drops into the E. coli and then 7 drops into the S. epidermidis test tubes. So I'm gonna take my scissors, cut this open. And we will use these 2 test tubes. So 1, 2, 3, 4, 5, 6, 7, and 7 drops into the E. coli, which is this one right here. 1, 2, 3, 4, 5, 6, 7. Okay. Next, I'm going to put this aside. As you can see, in the S. epi broth, it is fully red. It almost kind of gives you a fuchsia color. In the E. coli, it too is also red. So for the methyl red, both the S. epi and the E. coli did turn red. Next, I'm just gonna turn this, and next we're gonna go ahead and do the Voges Proskauer test. This is the epi and this is the E. coli. I am gonna take the Barrett's reagent A. I'm gonna cut that open and I'm gonna add 12 drops to each tube. So take our scissors again, cut that with the scissors. And we are gonna do 12 drops. So we're gonna start with the E. coli.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And another 12 on the S. epi. 1, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12.
Okay. Next, we're gonna just gently swirl that around. Put this down. As you can see, it's like a cloudy white. It looks like milk. Okay. So after that, we are gonna take Barrett's B reagent and we're gonna add 4 drops to each test tube. Okay. Let's cut that. 4 drops to each test tube. So 1, 2, 3, 4, and 1, 2, 3, 4. Okay. I'm gonna shake it gently for about 30 seconds, just so it mixes together. Okay, looks like it's mixed in pretty well. I'm gonna go ahead and let it sit for about 30 minutes. After 30 minutes, if there is a formation of some sort of red layer on the very top of this, then that means that it is positive. So we'll let that sit for 30 minutes and my results will be on my result video. Thank you.